the pattern of bone apposition and resorption distant sides. So stretching of the PDL is actually affects the tooth movement. It results in secretion of mediators, which mediates the osteoclast and osteoblast and results in tooth movement. Periodontium and bone responds to normal function. That's why tooth does not move during mastication. Even during mastication, we apply forces on the teeth, but teeth does not move. Why? During masticatory function, the teeth and the periodontal structures are subjected to intermittent heavy forces. This is the basic cause, right? If the forces are continuous, the tooth can move. If the forces are intermittent, even if they are heavy, the tooth does not move. The forces during mastications are usually for 1 to 2 seconds. The magnitude is from 1 to 2 kg for the soft food to 50 kg food. Chewing forces transmitted through PDL to the alveolar bone. So the PDL is very important for tooth movement. In case of dental implants and encalosis, tooth does not move. If you can see the table, physiological response to heavy pressure against the tooth. If it is less than a second, the PDL fluid is incompressible. As we know, the PDL ligament has. Can anybody tell me what is PDL space? What is PDL space? Can anybody tell me what is PDL space? Can you guys hear me? Yes. It is between the tooth and the alveolar bone, right? So PDL space where the PDL is actually present. The PDL has the ligaments, the PDL fluid, and the cells, right? Plus the blood vessels and the nerves. So the PDL fluid is incompressible if the pressure is less than 1 second. Alveolar bone bends and the piezoelectric signal is generated. And this alveolar bending is very important in maintaining the structure of the mandible, the normal structure of the bone, the skull. If the pressure is applied for 1 to 2 seconds, the PDL fluid expressed out because it cannot bear more. Tooth moves within the PDL space. That is physiological. That is considered as physiological. Even you can feel it when you clench your teeth hard during mastication. If it is for 1 to 2 seconds, the fluid expresses out of the alveolar bone. How? The alveolar bone surrounding the tooth is cribriform plate. What is the meaning of cribriform plate? It has pores. The fluid expresses out of the pores and the tooth moves within the PDL space. That is actually a cushioning action provided by the nature because of the PDL. If the pressure exceeds and it is between 3 to 5 seconds, the PDL fluid is squeezed out, the PDL tissue is compressed, and you will feel immediate pain if pressure is heavy. Why you feel pain? Because in that space we have nerves and blood vessels, right? If you have any question, you can ask during the lecture as well, please. PDL and bone response to sustained orthodontic force. Now, if we apply pressure intentionally, like in orthodontic treatment, heavy forces lead to rapidly developing pain, necrosis of cellular elements within PDL, and the resorption occurs is undermined resorption. But if the lighter forces, which are compatible with the survival of cells, are applied, it is painless and the frontal resorption occurs. Now, there are two different terms, frontal resorption and undermined resorptions. I will discuss them in the future slides. In orthodontic practice, the objective is to produce tooth movement by frontal resorption only because it is a healthy tooth movement. There are two types of theories in biological control of tooth movement. One is pressure tension theory. It is a classic theory of tooth movement. It relies on chemical signals as a stimulus for cellular differentiation and ultimately tooth movement. Blood flow is decreased when the PDL is compressed, while it is maintained or increased when the PDL is under tension. See how a tooth moves. Once the force is applied on the tooth, it results in compression of the PDL ligament, which is present in between, which is a soft tissue present in between the two hard structures, that is the alveolar bone and the tooth. Right? If you can see here, the PDL is present in between the two hard structures, the bone, which is cribriform, and the tooth. Right? So once the force is applied, what happens? The PDL gets compressed. So there is an area of compression, and in that area, once the force is heavier or sustained for more than 3 to 5 seconds, the fluid expresses out, the PDL compressed, the blood vessels occludes. Once the blood vessels are occlude, what happens? Hypoxia occurs. What is hypoxia? Less supply for the oxygen, which results in release of mediators, chemical mediators. And those mediators in turn activates the osteoclast. Osteoclast, that is the bone resorbing cells, which results in the bone resorption in the direction of the force. In the direction of the force. For example, if the force is applied in mesial direction, the area of compression is opposite. And the other area opposite to that, like the theory of Newton, the third law of Newton, in every force, there is an equal and opposite reaction. So, the area opposite to compression, the tension occurs because of the stretching of the PDL ligaments, which results in either same or increased blood supply, which results in activation of the osteoblast that forms the bone. So this is how the tooth moves. Once the force is applied, the area of compression is developed where the tooth moves, and the opposite to it is the area of tension where the bones is formed. Now, the effect of force magnitude, the heavier the force, the reduction in blood flow is compressed area PDL is more. The optimal force levels, the optimal force levels for orthodontic movement should be just high enough to stimulate cellular activity without completely occluding the blood vessels. So the aim in orthodontic tooth movement is what? Is the optimal force levels. What is an optimal force? It is the force in the orthodontic tooth movement which should be just high enough to stimulate the cellular activity without completely occluding the blood vessels. Because in the future slides, you will see what happens if the complete occlusion of blood vessels due to the force happens. So this chart is very important to understand the effects of the force magnitude, right? Physiological response to sustained pressure against the tooth. If the pressure is light, what happens within seconds, minutes, hours, and days? And what happens if the pressure applied is heavier, right? So if the light pressure is applied or heavy, initial 2 seconds are same. That is, the PDL fluid is incompressible, alveolar bone bends, which results in signaling known as piezoelectric signals. If the pressure applied, whether light or heavy, is for 1 to 2 seconds, the PDL fluid expressed out of the alveolar bone, the tooth moves within the PDL. That is physiological. Sometimes you feel it and sometimes you don't feel it. If the light pressure is applied for more time, that is 3 to 5 seconds, what happens? The blood vessels within the PDL partially compressed. Please focus on this. Partially compressed on the pressure side, dilated on the tension side, the PDL fibers and cells mechanically distorted. Within minutes, what happens? the force is sustained and you keep applying the forces, within minutes, the blood flow altered, oxygen tension begins to change, prostaglandins and cytokines are the mediators which are released, and within hours, metabolic changes occurs. Chemical messengers affect cellular activity, enzyme levels change. This is very important. In 4 hours, if the light pressure is applied sustainedly, it increases the cyclic AMP levels detectable. Cellular differentiation begins within PDL, means the cells which are undifferentiated will begin to differentiate into osteoclast, which in turn results in resorption of the bone towards the pressure side, compression side, right? And within 2 days, the tooth movement begins as osteoclast and osteoblast remodel the bone socket, the alveolar bone around the tooth. So this is happening if the light pressure is applied, which is the basic aim and desired in orthodontic tooth movement, which avoids the complete blood occlusion. But at the same time, if the heavy pressure is applied for 3 to 5 seconds, what happens? The blood vessels within the PDL completely occludes on the pressure side. Within minutes, the blood flow cut off to the compressed PDL area, and in hours, the cell death occurs in the compressed area. When the light forces were applied, metabolic changes occurring during chemical messengers, cellular activity was beginning, the cells starts to differentiate. Here,